So stem cells is a really important part of our study of aging and just body or somatic repair. But another thing scientists have to consider is how are they going to study it, and what criteria separates the different parts of their study. So one of the things is the turnover rate. Since stem cells are for every cell in the body, and of course every cell in the body doesn't replicate at the same rate. Some replicate at really high rates, some really slow. Neurons replicate really slowly, and blood cells replicate at a very fast rate. So I'll draw an arrow from here. That's to from here to there of the order of stem cell turnover rate from slow to very fast. I'll just give a few. So you got actually from fast to really slow. I could do that too. It's easier that way. So blood cells are very fast, and you got skin cells. And somewhere in the middle, you got something like liver cells, and the very slow, you have the brain cells like neurons, heart, and spinal cord. So when scientists are studying all of these, they are studying them individually. Okay, so they study them individually. The muscle cells are also called the satellite cells, and in muscle cells or any other stem cells actually, they notice that there is not really much a change in the stem cell number. Actually, in some cases, there is more stem cells as a person ages, but what decreases is the functionality because stem cells aren't immune to damage like any of the other cells. All cells can be damaged because damage comes in DNA mutations. You have non-degradable protein depositions that's building up in the extracellular matrix or the intracellular matrix or inside the cell as well. And then you have mutations in mitochondria DNA. And also you have mutations in the stem cell dish. So all of that combined together makes it work less efficiently. So that's why we say there's decrease in stem cell functionality. But actually, there's when you transplant the stem cell from an old patient to a younger patient, the stem cell functionality is renewed. So that is more evidence that says there is that aging is with stem cells is more due to the environment than the stem cell itself. At least how I think about it is that when each stem cell that divides, it may produce some somatic cells, some skin cells or blood cells that isn't perfect. They may have some mutations, and then as it produces more and more of those, like some stem cells give rise to more stem cells. Those stem cells give rise to more skin cells, and these skin cells that are produced with these mutations would cause the future stem cells to produce more cells with bigger mutations, or in a way that'll throw off the homeostasis of the niche, and then it builds up, just like the rapid oxygen species of the free radicals. The free radical damage to DNA builds up, and then when the DNA is damaged, it may not be able to fend off future free radicals, and it will cause even more free radicals to be produced. This is one idea proposed by Albie de Grey and many other scientists. So in a study of rats, when they joined the bodies of an old rat and a younger rat, miraculously, the old rat had health had better health. It combined. It's like it took the health of the younger mouse to renew itself. So that also shows that environment has a really big factor on aging. So, and then there's the notch signaling pathways. Signaling pathways are also important to how uh, stem cells respond to damage.